Hello everyone, my name is Emilio Balbuena and I'm a master's student in the Eroglu Lab. We're over at the North Carolina State University in the Plants for Human Health Institute. In our lab, we would like to uncover the molecular mechanisms behind the chemoprotective effects of carotenoids. Today, I would like to talk to you all about the project we've been working on regarding nutrient modulation of DNA repair in lung cancer. Lung cancer is the leading cause of cancer death worldwide, with 8 million annual global deaths attributed to direct tobacco use, according to a WHO report in 2019. In fact, 55% of lung cancer deaths in women and over 70% of lung cancer deaths in men are due to cigarette smoking. Cigarette smoking does increase reactive oxygen species, which increases the occurrence of oxidative stress, thus playing a critical role in the initiation, promotion, and invasiveness of lung cancer. In order to remedy this, a process known as base excision repair can be responsible for repairing most of the endogenous DNA damage, including oxidations and single-strand breaks. DNA glycolases, including 8-oxoguanine glycosylase, or OGG1, and neolike DNA glycosalases, are a family of enzymes involved in the recognition of base excision repair and this excision of DNA damaged bases. Regarding carotenoids, carotenoids are yellow, orange, and red organic pigments that give the characteristic color to carrots, pumpkins, tomatoes, and other fruits and veggies. These compounds happen to be produced by plants and algae, as well as several bacteria and fungi. The most popular carotenoid you've heard of is beta-carotene, the orange pigment in carrots, for example. As a whole, these compounds can be categorized as provitamin A, otherwise known as a vitamin A precursor, or non-provitamin A. These compounds have been observed to have antioxidant properties and exert important related biological functions, including scavenging reactive oxygen species. In our study, the objective is to explore the efficacy of all trans-retinoic acid, essentially vitamin A, beta-carotene, a provitamin A carotenoid, and lycopene, a non-provitamin A carotenoid, in cigarette smoking-induced lung oxidative stress, in order to illustrate the molecular mechanisms behind the chemoprotective effects of these nutrients. We propose that these carotenoids have further chemoprotective effects through the modulation of genome stability, like base excision repair. For the purposes of this presentation, I will solely be discussing the results obtained with lycopene treatment. This workflow depicts the process we underwent over the course of our experiments. First, we cultured the A549 human adrenocarinomic alveolar epithelial cell line in vitro. Following carotenoid pretreatment and cigarette smoke exposure, RNA was isolated for cDNA synthesis and quantitative PCR. Additionally, cell lysates were prepared for Western blot and oxyblot analysis. For this study, the A549 cells were seated in 6-well plates and pretreated with various doses of all trans-retinoic acid, beta-carotene, and lycopene. These treatments were 1 nanomolar, 10 nanomolar, 100 nanomolar, 1 micromolar, and 10 micromolar for 24 hours. Following the 24 hours, the samples slated for cigarette smoke exposure were introduced to the apparatus depicted here. The covers of the plates were removed and the open well plates were placed on the shelf inside. The filter of the research cigarette was removed and the remaining piece was inserted within a slot to the right of the apparatus. The cigarette was lit and then enclosed within the beaker. The smoke was kept inside and propelled inside the chamber via a fan, allowing for the cells to be exposed. The A549 cells were exposed to the cigarette smoke for a total of 45 minutes. The oxyblot analysis compared the levels of protein oxidation between non-smoking and smoking A549 cells. The left panel shows the band intensity of the oxyblot analysis, which depicts the presence of protein oxidation due to the detection of carbonyl groups formed from the oxidation reactions. The lanes here depict the levels of DNP hydrazone within corresponding concentration of lycopene treatments. The right panel shows the standardized quantification levels of DNP hydrazone due to the ratio of DNP protein and beta-actin control. The non-smoking cells do not appear to form any protein carbonization at all, while the smoking cells presented significantly higher levels of DNP hydrazone. It indicates that the formation of more oxidized proteins in the smoking cells. As portrayed in the figure, the quantity of carbonyl groups in the smoking cells were decreased with lycopene treatment at 1 nanomolar and 10 nanomolar, but not at higher dosages. Connexin 43 is a transmembrane protein that is responsible in part for the intercellular communication between adjacent epithelial cells via gap junctions. Gap junction intercellular communication plays critical roles in the tissue homeostasis and regulation of cell growth and differentiation. Cancer cells often display a loss of gap junction intercellular communication due to the dysregulated expression of connexin genes at multiple levels. Additionally, evidence has shown that tobacco exposure resulted in a lower expression of connexin 43. This phenomenon has appeared to have occurred within our study as connexin 43 levels seem to have decreased in the smoking cell samples. Connexin 43 protein levels was increased in the smoking cell samples with lycopene treatment at 1 nanomolar but not at other dosages. Nevertheless, even with lycopene treatment, connexin 43 protein expression was not restored in the smoking cell samples to the same level as the non-smoking cell samples. Such data could indicate that lycopene inhibits smoking-induced oxidative stress mainly through activating the base excision repair mechanism. Regarding the base excision repair mechanism, we checked the levels of the DNA glycosylase enzymes OGG1, NEAL1, NEAL2, and NEAL3. By further exploring the underlying mechanism that lycopene inhibited smoking-induced oxidative stress, we found that OGG1 protein expression significantly increased in the cells treated with lycopene at 1 nanomolar, 10 nanomolar, 100 nanomolar, 1 micromolar, but not at 10 micromolar. There was a trend of increasing OGG1 protein expression in the non-smoking cells with lycopene treatment at 1 nanomolar, 10 nanomolar, 100 nanomolar, and 1 micromolar, but the changes were not significantly significant. Consistently, our results show that in the smoking cells, NEAL2 and NEAL3 protein concentrations significantly enhanced with lycopene treatment at 10 nanomolar and 100 nanomolar. In the smoking cells, NEAL1 protein expression was elevated by lycopene treatment at all dosages, but the extent of increase was highest at 10 nanomolar and 100 nanomolar. Intriguingly, we found that at lower concentrations, lycopene treatment in the smoking cells promoted OGG1 expression to an even higher extent compared with the non-smoking cells. Our data indicated that lycopene initiated DNA repair more efficiently under oxidative stress, so we proceeded to explore the underlying mechanism further. SRB1 is responsible for the active process for the uptake of carotenoids, including lycopene. It has been shown that upon cigarette smoke exposure, SRB1 will go through subcellular redistribution and ubiquitination, leading to reduced cellular SRB1 expression. Interestingly, we found that the mRNA level of SRB1 was significantly increased in the smoking cells compared to that of the non-smoking cells, suggesting an increased carotenoid uptake in the cells under oxidative stress. In addition, we found that retinoic acid receptor beta or RAR beta protein expression decreased in the controlled smoking vehicle group compared to that of the controlled non-smoking vehicle group, but such decrease was attenuated with lycopene treatment in the smoking cells. In conclusion, we found that lycopene treatment at a lower dosage could inhibit cigarette smoke-induced oxidative stress and promote genome stability. As expected, smoking significantly increased oxidative stress, which was inhibited by lycopene as the antioxidant capacity was highest at 100 nanomolar. In cigarette smoking cells, lycopene increased OGG1 expression at concentrations of 1 nanomolar, 10 nanomolar, 100 nanomolar, and 1 micromolar, but not at 10 micromolar. Lycopene also improved NEAL1, NEAL2, and NEAL3 protein levels at 10 nanomolar and 100 nanomolar, suggesting that lycopene exerted an antioxidant effect at lower dosages. In addition, lycopene treatment restored the connexin 43 protein expression at 1 nanomolar. Intriguingly, we found at lower concentrations, lycopene treatment in smoking cells promoted OGG1 expression to an even higher extent compared to non-smoking cells, indicating that lycopene initiated DNA repair more efficiently under oxidative stress. And of note, all trans retinoic acid and beta-carotene did not show any antioxidant efficacy in our study. And that's it. Thank you everyone for listening. I'm really glad that I had the opportunity to share our results from our study. We actually have a manuscript in the works, so hoping to get it published soon, so stay tuned for that. Thank you.